Our next speaker, may I call upon Mr. Manoj AM?
So, good afternoon to one and all, and thank you, BASAI, for this opportunity. We are from Tea Stains and Company Limited, and the topic we are going to present here is on the role of microbes in the crop protection and nutrient segment. Even though the list is very longer, I'll not take much time here. Coming into the I'll give a small brief about the company. It's a 160 years old company of Indian origin. We are into various segments of agrochemicals, starting from biopesticides, biostimulants, biofertilizers, etc. And we are developing our new products through our R&D center, which is DSAR accredited R&D center, and we have a global presence in 32 countries as of so far. Directly coming into the topic, generally microbials we have classified into two segments, either as crop protection or crop nutrition segment. Here we will speak much on crop nutrition segment, as we are all more familiar on the protection aspects. First, I'll start with biostimulants. We are all speaking about biostimulants for the past 2 days. One common thing, this is a list of countries where it has been already defined a definition for biostimulants. The common thing what we can observe in all the definition is that it's one thing is that it is an product which is a substance or a microorganism or a mixture of organisms which helps in enhancing the tolerance effect, nutrient uptake, or the efficacy of the crop. That is the quality of the crop. That is the one common thing which is being spoken by all the definitions across the globe. Coming into the abiotic factors, already microbials have been proved for its abiotic stress factors. There are like plant growth promoting rhizobacterias and plant growth promoting fungi, which has already proven for its effects. It is being achieved through various roles, which we call it as strategies kinds of things. Like they produce phytohormones, siderophores, they lower ethylene levels, they regulates the genes and antioxidant genes. These all plays a major role in the abiotic stress management. We have taken few examples, like I'll say with one or two. If you take Pseudomonas, which produces IAA siderophores, which has benefits of its growth towards in the stressed plants. It increases the potash availability, sequestration of materials, beneficial effects of rhizobacteria. And these are all the list, like arbuscular mycorrhizal fungi, which increases the uptake of K plus, Ca2 and Mg2 plus, which helps in improving the plant growth or biomass production in the drought tolerance plant. So what we can see here is a microorganism as such has a role in the part of abiotic stress management. Coming into microbe, till now we were seeing about microorganism as single compound. Microbiome is a combination. It was first derived by Ledberg and McRae. The most important factor in microbiome is we need to understand about the plant to plant interaction, microbe to microbe interaction, and a plant to microbe interaction, because as we produces reflex effect when we attain into some difficult positions, plant also produces certain organic compounds or volatile materials which helps in protecting them from the stress factors. Few like antibiotics, volatile molecules, phytohormones, organic acids, amino acids. These are all the few compounds which they produces during the stress factors. Generally, this microbiome is classified into three. Either it is a rhizosphere, endosphere, and phyllosphere. It's very simple based on their classification. Rhizosphere, which is below the soil, near the root zones. Phyllosphere, which is above the soil, on the leaf surfaces. Endosphere, which is within the plant surfaces. So I have tried to list down what is the role of these rhizosphere. Like the rhizosphere microbiome has a role in pesticide degradations, IAA productions, and it has been reported for various activities. Like endosphere, we all know it has nitrogen fixation, phosphate solubilization, all biofertilizer effect we can see with endospheres. And phyllosphere also we have drought tolerance effects, antagonistic activities. These are all being produced by the phyllosphere microorganisms. So the next aspect of nutrition is on biofertilizer, which is in India. If you take in India, it is confined with N-fixers and P solubilizers or nutrient mobilization, and it is also limited with few organisms, like which is listed here, and the which is one which is bolded underlined is like it is classified as a biostimulant in other countries, which we were discussing for the past 2 days. And as we had in the earlier meeting also, like it is limited with few organisms like Astobacter, Azospirillum. These are all the organism which is recommended for the end fixing products. But if you see something which is outside India, the products there are like Encieria technology, which is an end fixing bacteria, but it is not of the four listed. It is a different one of Gluconobacter. You see Blue N, which is an end fixing bacteria again. It is of different origin of Methylobacterium. So there is an opportunities of having microorganisms role. And the limitations what we see here is in India, it is we are limiting the organisms based on the classifications. For example, a Pseudomonas, which is already being recommended as an pesticides, even though we have seen that there is a property of its biostimulant effect, it is not a biostimulant or a biofertilizer. It is classified as biopesticides. And EU, it is already stopped with 4 actives so far, like communications like EB likes of committees, they are even opposing with more number of microbes, but still as of date it is only 4 listed actives are being called as microbes there. And with US, UK and other countries, it is not fully defined for microbial based products. And what we expect is that if the microbes possess an biostimulant effect or a biofertilizer effect, which can be proven through a molecular level or an efficacy level studies, it should be considered for the regulation aspect. And the scope what improves is that we'll have a sustainable crop growth, we have an enhanced tolerance effect, we can help in the reduction of the pesticides loads. And the process how we develop a microbial biostimulant at tea stains is first we'll isolate an microbe, we'll do a complete characterization in terms of its basic features, we'll submit it to the regulatory bodies like NBA, NBAM, and we'll obtain the clearance. We start with lab clearance screening like we do with various pathogens, insects, biostimulant traits, and we see on what character, like whether it produces siderophores, what phytohormones it produces. And once when we are clear with laboratory level, we take it up to the semi-field level where we do potted or greenhouse conduct kind of evaluations. Then it later goes to the field evaluations. Once when we are clear that the product is suitable for an field level market, then we standardize the method of production and we take the product to the bulk level. And in tea stains, we have launched latestly with 2 microbial biostimulant. It is not into the market. We have submitted it for registration, but we have obtained a patent for this product. It's a patented microbial biostimulant. It has a number of properties. We have consigned with 2 properties here. It is one is it produces siderophores, which is most important in the growth, and second one is it helps in the root colonization, which in turn helps in the water uptake, nutrient use efficiency. And this product we have tested across India, and it has proven that it works across all soil, geography, and all climatic condition. It has proven to have the higher efficacy due to the even at the abiotic stress condition. This is just a glimpse of trials what we have conducted across India. We have started from Tamil Nadu up to Rajasthan. We have conducted trials, and starting from paddy to sunflower, we have conducted trials at various crops at various locations. And one thing what we have observed is amongst the different number, it produces yield increase in all the crops what we have obtained so far. That is a single microbe based product, and this is a microbiome based product which we developed in coordination with an ICR institute. Here we have co-cultured around 10 microbes for its bioefficacy, for its bacterial, it's a 10 bacterial microbiome. And the main role is that it helps in the silica solubilization, which helps in the abiotic stress tolerance, which we have also conducted studies to an reputed ICR institute, and we have identified that these abiotic stress genes are getting activated when you apply this product on the crop. And this product was also tested by various locations, and we have found it has been effective across all the crops. And this is our overall biostimulant microbial packages what we have so far. We have microbial biopesticides, and we have biofertilizers, and currently we have 2 microbial biostimulants which is under regulations. Thank you.